This is the fungi lab. So you can put that at the top. What you're going to be doing in this lab is learning the life cycles of the 5 fungal phyla, the 5 different groups of fungi. Just to cover the classification of fungi, fungi are in the domain Eukarya, just like the Protista that we studied last, the protozoans and the algae. Eukarya contains the Protista, the fungi, the plants, and the animals. So we're in the domain Eukarya, so the cells are eukaryotic. All the cells have a nucleus and organelles. And then the fungi is the kingdom. So we're in the kingdom fungi. And then we have 5 phyla. Phyla is plural. We would say, like, for example, phylum Basidiomycota. That's the first one that we're going to cover, the life cycle. So there's the phylum Basidiomycota. There's the phylum Zygomycota. Now, the Basidiomycota, just so you can kind of get relate these to things that you know, the Basidiomycota are your mushrooms and your puffballs. The Zygomycota is your black bread mold, mold that grows on bread. That is Zygomycota. And then the Ascomycota is definitely the largest and most diverse of the phyla. It's going to include your baker's and brewer's yeast, the yeast that's used to make bread and beer. It's going to include penicillium, which is used to produce the antibiotic penicillin. And Ascomycota will also include truffles and morels, which are edible fungi. Okay, so the Ascomycota, then there is the Glomeromycota, and the primary fungus that we learn about in this phylum is the one that forms a symbiotic relationship with plant roots. It's called mycorrhizae. So Glomeromycota is going to be mycorrhizae, and then our last group is going to be the Chytridiomycota, which tend to be parasites. They tend to cause disease. Okay, so these are our groups, and what you're going to do in lab is you're going to copy everything you see me write. And this is not how lab would be typically. We would typically have some hands-on activity, and you get to look at some specimens and that kind of thing. But with our situation that we're in, I've seen other instructors do this, and I think it helps when you can. I've learned from watching my children too, taking classes online. I think it really helps when you draw processes out. When you draw them, it really helps you learn them. So we're going to start. I didn't finish. So what you're going to do for this lab is you're going to take the same notes that I give you. You're going to draw everything that I draw. You're going to take pictures of it, and then you're going to make 3 drawings of your own, 3 life cycles of your own, and that's going to be the life cycles of Ascomycota, Glomeromycota, Chytridiomycota. But everything else is going to be copied. I'm going to draw it, and you're going to copy my drawings. And I'm not a good artist, so there's no real how do you say it? High expectations. You'll see if you haven't seen my drawing yet. But anyway, so I'm going to go ahead and do that. By the way, you're going to take the pictures. You're going to put them into a document and upload them to the assignment. Okay, so just to show me that you did it, and then you did the 3 life cycles for Ascomycota, Glomeromycota, Chytridiomycota. So I'm going to start by drawing a mushroom, which is your example of the Basidiomycota. Write this a little larger, just so you can understand when we do learn about the life cycle of the mushroom, just so you can understand what the structures are, where they're located within the mushroom. The mushroom itself is called the basidiocarp, and it's going to have a stalk. has a collar on it called the annulus. So this is the stalk. This is the annulus or collar. And then the cap of the mushroom, just dome shaped, but we also want to draw the gills. So we'll come out here like this and try to draw three-dimensional. I've practiced this a little bit, which is sad because this is the best I've come up with. But this is the pileus. The cap is the pileus. Then the gills are called the lamella, and lamella simply means layers. You can see these gills are layered. There's also lamella in bone tissue. So lamella is a very common anatomy word that you see, in human anatomy and other organisms. So the gills are the lamella, which just means layers. Now, this whole structure, this mushroom, oops, that was not pretty. Okay, that's supposed to be an arrow. Sometimes you make it worse when you keep on. That looks like that looks like a little tiny mushroom, doesn't it? But anyway, the mushroom itself is the basidiocarp. Basidiocarp. And what's going on is the basidiocarp is the reproductive structure. It is the structure that produces the spores, and the spores are going to be they're what's going to land on the substrate, usually the forest floor, and grow into a mushroom. So within these gills are going to be structures called basidia. Individual is basidium, but these basidia are going to produce the spores. The basidia will produce the spores, and the spores in the basidiomycota, the spores have a special name, basidiospores. Okay, so now we're going to draw the life cycle. And we're going to start our life cycle with the basidiocarp, with the mushroom. We're going to draw it down at the bottom of our page. So we've got to do this again, except I'm not going to be so particular. Oh, I didn't even talk to you about the structures that are growing down below. Sorry. They're thread-like structures. They look like roots, but it's actually called, all together, these threads are called the mycelium. But an individual thread is called the hyphae. And all fungi produce hyphae. They grow thread-like structures. The mushroom is just the fruiting body. The largest part of the organ, or of the mushroom body itself, is the mycelium underground. And you may have learned this as a child. But this is our stalk, and we'll draw our annulus like that. And then, again, we've got our cap. Drawing it a little faster this time. We've got our gills underneath. And we're going to start with, since we know that the gills produce spores, we're going to start with what happens is that a zygote is produced from this mushroom. And the zygote is going to become the basidium, and the basidium will produce the basidiospores. So this mushroom produces a zygote that is diploid, and it's just a single cell. And I'm going to put 2 in. Remember, that means diploid. That means it has a chromosome. Doesn't really have a father and a mother, or a male and a female, but it has 2 different mating types. So the zygote is diploid, meaning it has, for every pair of chromosomes, one came from the positive mating type and one came from the negative. And since the nucleus is diploid, what we're going to do is use blue and red for the nuclei of the positive and the negative mating types. So when blue and red combine, you get purple. So I'm using purple for the nucleus of the zygote. Now, this zygote is going to undergo meiosis. So you should remember a little bit about what's going to happen. What should happen with meiosis is your diploid parent cell should divide into 4 haploid daughter cells. But that doesn't happen right away. What happens is the nucleus of your zygote does divide, and so you get 4 haploid nuclei, but the cell does not divide. So this grows into the structure we call the basidium. And what the basidium is going to do is going to undergo cell division, just cytokinesis basically. And it's just a separate process. And it is going to end up forming 4 basidiospores, which are haploid, of course. Okay, so these basidiospores are going to land on something suitable for growth of the mushroom, suitable for the mushroom to germinate, and so moist soil or somewhere where there's decomposing organic material, that kind of thing. But they're going to undergo dispersal and germination. And let's label these. Sorry, basidiospores. They're going to undergo dispersal and germination. And then they're going to form into 2 different mating types. And this is we're going to start drawing the hyphae that combine together to form the mycelium. So we're just going to draw a few, but the cells kind of link together. So we're going to draw the positive mating type first. And then a little cell here. And the positive mating type we're going to give red nuclei to. So remember, these are all haploid cells. So this cell in particular is going to mate with or fuse with the same cell on a negative mating type structure. So we'll draw now the negative mating type, which we're going to use blue. For the nuclei of all of these cells. And all of these came from this, they're all still haploid. They just came from the division of the basidiospores. Some become the positive mating type and some become the negative mating type. Then something's going to happen that we haven't really learned, that we didn't really learn about in Bio 111. And we don't normally talk about it that much in Bio 111. And it's called plasmogamy. Plasmogamy is the fusion between the positive and negative mating types. And all the cells that come from that are going to have 2 nuclei in them. So they're going to be dikaryotic. So we're going to end up having growth from here. Once these 2 fuse, you have to kind of picture that in your mind. But we can just draw it over. All right, so we're going to end up having, let's draw our positive mating type. I think I drew 5 cells and then 1 cell here. Draw it a little longer this time. And then okay, so we've got our negative mating type. We have the blue nuclei. And then our positive mating type will have the red nuclei. And then when these 2 cells fuse together, we call that plasmogamy.